Yesterday in two panels, we learned about the current standard of care, treatments that are available and how to make the most of those. And then in the second panel session, we learned about treatments that are already available, but not standard of care. And they're the treatments that are in clinical trials or can be prescribed by an expert. So today's session is actually the one that I'm most excited about, because this is the one that is really our future. What TKIs were 15 years ago. And, you know, this is what the fourth generation TKIs were 6 years ago. These are the things that are just coming up now. And they're going to be coming into clinical trials shortly, or sometimes they're already in clinical trials, but not including outpatients. On the panel today, we've got Dr. Mark Muscovich. Mark was diagnosed as an outpatient in 2020. He was a professor for I don't know how many years, teaching cell and gene therapy, so he's no dummy. We've got Dr. Emily Venanzi, who is also an ALK-positive patient. She was going to be here in person, and she didn't get COVID, but her kid got COVID. So she didn't want to bring it here with us. And so she's going to be here remotely. Dr. Muscovich, I'm going to call him Mark from here on. And Dr. Venanzi, going to call her Emily from here on, going to do a presentation. And I've given them very, 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 very strict instructions to make sure that everything they say could be understood by a 9-year-old. Because I know yesterday was, you know, impressive, but it was over even the heads of people that were science people, much less the people that are not science people. We also have with us Dr. Erin Schenk from University of Colorado, Dr. Vincent Lamb, and Dr. Ben Creelan, and they are all cutting-edge researchers. So with that, we're going to hand it to Emily and Mark to make the presentation in words that all of us can understand. And then the panelists are all going to discuss these treatments, again, in words that everyone can understand. But there's, you know, we're talking with companies, we're talking with the CEOs and the chief scientific officers, we're talking with the world's leading researchers, and it really is exciting what's being developed. Really, really is.
Okay, thank you, Colin. So good morning, everyone. Good to see you. Good to see you here. I might aim for 12-year-olds instead of for 9-year-olds, but great. And Emily is on the screen. You can see her there. Good morning, Emily.
Hi, everyone.
Hi.
Wish I could be there in person.
Right. So Emily and I are charged with giving a brief background. And so if we go to the first slide, that might be the title slide. And if so, we can skip to the next slide. So there are a lot of words on the slides that we've prepared. Don't worry about reading the words. Listen to the sounds of our voices. And you can look at the words afterwards, because these present decks are going to be posted online. So let me give you a really brief background that is going to be useful, I think, because it will help us talk about the therapies and think about the therapies we want to discuss. So it turns out our lungs normally get worn out. So breathing oxygen is actually very stressful on our lung cells, which means our lungs are normally dividing. The cells in our lungs normally divide and replace themselves because exchanging oxygen and CO2 is stressful. It breaks the parts of your cells, and so you have to make new cells because your old cells are broken. And so when in those cells, the ALK kinase gets turned on because some genes have gotten rearranged, which is just good, bad luck. It's just good, bad luck. That's what I tell people that I have. Because I'm ALK positive, I have good bad luck because I have targeted therapies, right? But when ALK gets turned on, it tells those cells that are normally dividing to divide even faster. And the faster a cell divides, the more mistakes it makes. And then eventually it accrues enough mistakes that it does two things. It gets out of control and it becomes a cancer cell. And it evades your immune system, because all of us would have tumors all over our bodies all the time if our immune system weren't normally killing cancer cells as they arise. So pat your immune system on the back, because you only have the number of tumors you have right now. I mean, we wish we had none, but in fact, your immune system is still doing a job in many places in your body. So if we go to the next slide, then we have our targeted therapies. So our targeted therapies actually inhibit that ALK kinase. And so that signal is telling our cells to divide even faster in the lung. The ALK tyrosine kinase inhibitors inhibit that signal. So now they just go back to the normal level of replacing our cells instead of being in overdrive, right? And these are a set of gene rearrangements, some of those that turn on the ALK kinase in those cells. So then on the next intro slide, and the last intro slide, what happens? How do we treat ourselves? We have these tyrosine kinase inhibitors. They turn down that signal. They block that ALK signal that's been mistakenly, because of our good bad luck, turned on in those lung cells. And that can slow down the signaling so your immune system can catch up. Or if we can stimulate the immune system, it can also catch up with cancer cells and kill them. And so our tumors will shrink, right? Of course, cancer is intelligent when you anthropomorphize it, right? And so when you get mutations in the ALK kinase, so the inhibitor can't bind to the kinase and stop it anymore, now the kinase gets the upper hand again, starts telling your cells divide, 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 and cancer is back, progression. And sometimes, as we've talked about, instead of the ALK signaling pathway happening, another signaling pathway gets turned on so the cells can keep dividing. That's the bypass, right? And so some of these other drugs we take, and Emily's on a combination trial, for instance, they will block one of these bypass pathways. So now, even though we stopped ALK, we can stop the bypass and again, slow down the cancer cells enough for immune system to catch up. So I wanted to give that background. So if we go to the next slide, then this is the backdrop for our presentation. So there are a lot of reasons to hope because there are many, many therapies in the pipeline that some people are already sampling in clinical trials, and some people will soon be able to sample in clinical trials. And we categorize these on the slide. We're really going to focus on a subset today because we only have a half an hour. And so we're really going to focus in two areas. One is stimulating the immune system. So stimulate the immune system to catch up with cancer and kill it, right? The other way is new small molecule therapies like these tyrosine kinase inhibitors, but new molecules that will also attack the signaling system, either the ALK system or the bypass system, and turn it down. So again, your immune system can catch up with the cancer cells and kill them. All right. So then I want to hand it over to Emily, and she's going to talk about the immune stimulation approaches very briefly. And then I'm going to speak about the small molecules briefly. And then we're going to have a discussion with the true experts about, what is on the horizon? What are they confident about? What are they excited about? And then we'll have a chance for you to ask questions. So Emily, you want to take it?
Yes. So if you can show the next slide. As Mark already said, the ALK fusion protein is expressed in tumor cells and it drives cancer growth. But ALK is expressed very little in other parts of the body. And in fact, it's nearly absent except during fetal development. So it could potentially be a really good immune target, one that's specific to tumor cells. So one potential way to drive an immune response to ALK is by using vaccination. This is a therapeutic vaccine given after the cancer has already been discovered. So Dr. Chiarli spoke yesterday about his research showing that an ALK vaccine can lead to an anti-tumor immune response in mouse models. And his group, as well as Dr. Lamb's group, are now working on ways to bring this approach to patients. Next slide. Most ALK lung cancer tumors are what's referred to as cold tumors, meaning they don't seem to attract the attention of the immune system. One way of turning an ALK tumor from cold to hot, making it more attractive to the immune system, may be to use a type of virus called an oncolytic virus. And these viruses are made specifically to infect tumors or to otherwise provoke the immune response to attack tumor cells. And there are multiple companies, some of which the Research Acceleration Committee has already met with, working on these types of viruses. And these work by either killing tumors by infecting them directly, causing inflammation in and around the tumor, or causing tumor cells to make immune stimulating factors. Next slide. Another way to enhance the anti-tumor response is by using genetically engineered T cells. So T cells are immune cells that kill other cells in the body when they don't look the way they should. So for example, if they've been infected with a virus, or potentially if they're cancerous. There are researchers working on designing T cells so they can specifically attack cancer cells. And the idea is to create a large number of these engineered T cells in the lab, using a patient's own T cells as the starting material, and then infusing these back into patients, where they would then go and attack the tumor. Dr. Gaynor and Dr. Chiarle both talked yesterday about their work on finding ALK-specific T cells in people, and then using the receptors on these T cells, identifying them, and then potentially using them for this type of therapy. Next slide. Similar to using engineered T cells, another cellular therapy already in clinical trials involves isolating T cells called tumor infiltrating lymphocytes, or TILs, from a surgically removed tumor sample. These TILs are then multiplied many-fold in the lab and then infused back into the patient. The hope is that these TILs, infused in large numbers, will be able to attack and eliminate the tumor cells. Next slide. We also heard a little bit yesterday about what's called the tumor microenvironment. So all the other cells, blood vessels, and molecules in the area of the tumor. There are new therapies in development that aim to modify the tumor microenvironment to make it more immune stimulatory and less friendly to the tumor. So again, turning a cold tumor hot. Next slide. One way that tumors keep the immune system from attacking is by turning on so-called immune checkpoints that tell the immune system, stay calm, there's nothing to see here. Most of us have heard that checkpoint inhibitors that block the PD-1, PD-L1 pathway are usually not effective for ALK lung cancer. But there are a number of other checkpoints that researchers are working on targeting for cancer therapy. So that was a very brief overview of some of the exciting research going on to try to harness the immune system to treat ALK-positive cancer. Now Mark will go over some of the other types of potential new therapies in development.
Okay, thank you, Emily. I'm only going to talk about two therapies, and then we're going to open the discussion. So one is that the TKIs work by inhibiting the ALK kinase from putting phosphate molecules onto other targets and sending signals that say to a cell to divide. But another way to stop the ALK kinase would be to destroy the protein, to actually degrade the protein. And so if we go to the next slide, the idea is that if we could make a molecule that would bind to the ALK kinase and draw it into a place where it could be destroyed, then we could destroy the protein and stop the signaling. So on the next slide, it turns out our cells on the next slide have recycling bins. And they're called proteasomes. And so when a protein gets old or damaged, there are other proteins in our cells that actually tag that protein with something called ubiquitin, because it's in all our cells, so it's called ubiquitin. When you tag a protein with ubiquitin, it gets sent to the recycling bin and it's chewed up and destroyed. So if we could send ALK to the recycling bin in the lung cells, we'd stop the signaling. And in fact, Dr. Barpeled and Dr. Ifredi's work yesterday is about exactly that. They were talking about small molecules that may actually drive ALK kinase in lung cells to the recycling bin and destroy it. And so these protein degraders, as they're called, because they cause the degradation of the ALK protein, are a new and exciting approach. And then on the next slide, in addition, we've talked about the fact that when ALK kinase is turned on, actually it signals by multiple molecular pathways. There are a lot of handoffs of signals that tell cells to divide. And on the next slide, it turns out that there are all sorts of different ways that the cancer cell will turn on other ways to signal cells to divide and evade the immune system. Those are the bypass pathways. Now, the challenge is that those bypass pathways have their own little relay pathways. And so if we wanted to attack each pathway, we'd have to find one inhibitor for pathway 1 and one for pathway 2 and one for 3. But on the last slide, for this intro, those red X's all cross out a protein called STAT3. It turns out multiple bypass pathways use a protein downstream of the receptors that turns on the signal called STAT3. And there are companies making inhibitors that inhibit STAT3. if you can inhibit STAT3, you may be able to actually shut down multiple bypass pathways at once. So if you could take a STAT3 inhibitor and combine it with the TKI, you might be able to shut down not just the ALK pathway, but the bypass pathways and multiple pathways at once. So if we go to the next slide, this is the backdrop for our discussion. We've talked about the immune stimulating approaches on the left. Emily's talked about that. I've talked about a couple of these molecular approaches on the right, but we wanted here to throw the discussion open to the panel and say, what are you excited about? And are there other therapies that we haven't discussed because we couldn't discuss everything? Colin gave us 6 minutes and I didn't clock myself. I think we probably ran over a little bit, but I think we still have at least 30 minutes plus time for questions. So let me throw it open to the panel.
I want to say, I know that went down really, really well because Nancy was sitting there like she understood it. So. I guess the first thing I want to say is that when I look at the presentations by these different companies and researchers, a lot of the preclinical data is looking really exciting to me. And so I'll ask the experts here, what sort of preclinical data have you seen for what types of treatments that really get you excited?
I guess I can go first. First off, I really want to thank everybody for making time to join us this weekend. It really is an honor to speak in front of you and to be part of this organization to help advance research, and that's what drives us every day. And again, it really is such a delight to be able to speak on this panel. I think Mark and Emily did an amazing job summarizing some of the key areas of what we're looking at in terms of the up and coming and emerging technologies and therapies that may be coming down the road that are applicable to ALK. In terms of some of the areas that I think that are most promising, I do think the preclinical data for the protein degraders is actually getting more mature, and it looks like it's something that actually could be applicable to the ALK fusion protein, although there's still a lot of challenges and hurdles ahead of that. But that's one area that I'm pretty excited about and sort of keeping a close eye on. Of course, I'm a little biased in that our focus at Hopkins is often really with the immunotherapy approaches. And so just trying to do a lot of work to lay down exactly what the immune microenvironment that Emily had mentioned looks like in terms of specifically about the T cells and what state they're in and why we don't have as good of a immune sort of response in ALK. But also, very importantly, I think we're starting to understand that it's not all about the T cells in ALK, as it is with other cancers. That we're seeing the same thing. It's actually about the other cells that make for a hostile microenvironment such that the T cells aren't able to be as effective as we would like them to be. So I think the strategies to first better identify what's going on in the microenvironment are really important. And then the ones that have been highlighted in terms of targeting macrophages and other cells of the microenvironment are going to be really important in terms of helping make cellular therapies or vaccines work as well as we'd like them to work. And I know Ben's done a ton of work on the cellular therapy front.
Yeah, I think to that point, in immunology, we tend to be T cell chauvinists, meaning that our research has been exclusively focused on the cell type that has been well known to destroy viruses, bacteria, and even tumor cells. But there's a whole other side of the equation, which is what actually stimulates that T cell to work and tells it what to do and directs it. And so more work on macrophages, B cells, and dendritic cells, which are the antigen presenting cells, the one that actually presents that ALK protein to the T cell, is really important and poorly understood. So some of the methods that were described in the last slide do address that side of the equation. And again, that is understudied. Right now, our focus has been in T cells, right? The killer T cells. And that's what a lot of that cell therapy you saw on the previous slide was, whether it's some of these drug companies like Instill Bio, Iovance, or other CAR T cell or TCR type companies, which are much more in their infancy. Those are options for patients. So the cell therapy that probably is the biggest option for patients right now would be general TIL, where we just resect a tumor, grow it in culture, and then hope that there's plenty of T cells there that actually will attack the tumor. But the problem is we don't know whether it's actually targeting the tumor when we give it to a patient. It's really just we find out afterwards.
Going back to your question, Colin, and again, like Dr. Lamb said, I'm really delighted and honored to be invited to speak with everyone today. One of the preclinical pieces of data that I saw recently that really drew me in was a group from Harvard using the Don't Eat Me pathway, the anti-CD47 pathway, where in animal models, they gave antibodies that block that pathway to animals bearing ALK-positive tumors, and they saw improvement in control with targeted therapy. And I think, getting to that point where we can even more effectively eradicate some of that residual disease. And maybe that's by, stimulating that component of the immune system that sees the cancer cells and helps to pull them up and pull them in and destroy them internally, is a really fascinating approach to me. And my bias, of course, is the tumor microenvironment and the monocytes and the macrophages that are within the cancer mass, because it's more than just the cancer cells. When your doctors and teams talk about nodules or tumors or masses, it's cancer cells and everything they've done to try to make a really good place for themselves to grow and survive in the body. So that's blood cells. That's convincing immune cells to come in and protect them from those attack T cells and really help foster an environment where they can grow and survive. And some of the things I've been really interested in within translational research, as well as in my laboratory, is trying to eliminate one of those helpers, one of those macrophage monocyte subtypes that really help to support and protect the cancer cells from T cell attack. Because just like Dr. Creelan was talking about, there are many different ways cancer cells can avoid immune cell attack. Getting other immune cells to protect them is one way. It's sort of like they're the bouncers in the tumor microenvironment, perhaps. They're the big burly guys protecting the cancer cells from the T cells.
What are the aspects that you're most excited about, Mark?
Well, I think that because, as I said, the reason we don't have, and our relatives and friends don't have tumors all over our bodies, is because our immune systems are really good cops, usually. They usually take care of cells that get out of hand. And so I think that immune stimulation is our natural mechanism. Most of us don't make TKIs in our liver or anywhere else. And so, I think immune stimulation is the natural approach. I think so I'm most excited about that, but I love my TKIs and I hope they're going to last me a long time. I'm 2 years in on alectinib, so, that's great. But we all hope they're going to give us long runs as long as possible. I think immune stimulation is great. The other thing, just looking at it as an aside, is just in case you're wondering, there are at least 30 distinct different types of cells in your immune system. So when you hear about Tregs and natural killer cells and macrophages and monocytes, so don't be concerned about that. There are lots of different ways. And so the point is that there are many different approaches to trying to attack the tumor. And the problem is that tumors are there because they have evaded normal surveillance. And so Aaron was talking about, some of the cells, the bouncers, protecting the tumor from other cells in the immune system. So, we call cancer intelligent, but even if it's not intelligent, it's unfortunately successful in us, right? And so we need to try to frustrate its success and reverse it. But I'm most excited about the immune approaches. Though small molecules probably have a simpler path, and we know better how to approve those through FDA. Cell-based therapies, vaccinology is moderately well understood. Cell-based therapies is a new area with FDA, and that's going to be more challenging to set that up, though those therapies are in development, are out in the clinic already in some instances.
We don't want to forget you, Emily. Which is the technology that you're most excited about? Personally, for me, the data that I've seen so far, oncolytic viruses, and I would love for the panel to talk a little bit more about oncolytic viruses in just a little bit, but I'll just have Emily talk about what she's most excited about, and then I can tell that we've got Ben's really excited to get onto that.
Well, I'm with Mark. I think the immune-based therapies have a lot of promise. And the aspect of the immune system that I think is very interesting to think about is that it has memory. So when there are T cells in your body or other immune cells that are able to recognize the ALK expressing tumor cells, they'll do that now, and they'll also do that 20 years from now. So the potential for a long lasting treatment is there with immune-based therapies that I think is a bit different than what we have right now with the TKIs.
That's a really exciting point. So we all realize now, I think, when TKIs first came along and we'd see a lot of patients go NED and you're like, wow, we found a cure. Well, we realized after a few years that the cure is temporary. So a lot of the things that are now under research, I think, have the potential to truly be something that makes it 10, 20, 30 years, and by which time there'll be something even better. So the thankful thing for all of us is that TKIs is giving us time to see these things getting developed. So that's what's really exciting. I love the passion that these researchers bring. They just want to get the microphone to their mouth and start talking. So go ahead.
If I may, just a quick point to Emily's point about the T cell memory. One of the exciting things that we're starting to see, and this is again very preliminary, is that in the blood of ALK patients who are actually doing well on their TKI, often without any significant cancer on their scans, we're actually able to detect significant T cell activity in their peripheral blood. And I think Justin Gaynor may have presented some of this data in his update yesterday. So that's really exciting to us. It tells us that that there is some immunogenicity associated with ALK, which traditionally has always been a big question because ALK is not very irritable to your immune system. It looks so similar to normal cells. And so to see that there is some T cell activity, that gives us a lot of hope that potentially we could actually further enhance that activity with a vaccine or cell therapy or any of these other immune-based treatments.
I totally agree there. I think it's underappreciated, the immune stimulatory capacity of starting targeted therapy, and in terms of what your body does to initiate that elimination of the cancer cells. So I think there is a window of opportunity there to really harness sort of the normal natural immune response that patients can generate post-TKI.
Well, to your point about oncolytic viruses, so it's actually quite promising because when you do inject these oncolytic viruses into tumors, and that's where most of these trials have started, you have to have an injectable tumor, right? So safely accessible by a needle. And we do see that the virus does a great job of wiping out the tumor where it gets injected, right? But the problem is trying to turn that into a domino effect where it actually stimulates your immune system or other things to destroy other tumors outside of where it gets injected. And I think that's the next big hurdle for oncolytic viruses, is trying to get what we call the abscopal effect.
I think the data that I've been seeing on my studies, and Mark would have had a better understanding of this, and Emily would have had a better understanding, but we had meetings with companies, and My studies were showing really got everywhere, including the brain.
Systemic virus, right?
Yeah. So
Yeah,
Mark can explain it better than I can, or
yeah.
Emily even probably better.
Emily, do you want to talk about the mouse model experiments and the abscopal effect with the oncolytic viruses?
Sure. So some of the
And Emily's going to explain what the heck abscopal means.
So yes, first I'll try to define that. So abscopal, and correct me if I'm wrong here, but involves treating one specific tumor site and having that immune response at that site then spread to other sites of cancer in the body.
That's correct.
And this is talked about sometimes with radiation, where and it's controversial whether it actually happens, but radiating one lesion then leads to other lesions being eliminated by an immune response. But the same idea is there with oncolytic viruses, that perhaps targeting one tumor area, one particular lesion with an oncolytic virus could then rev up the immune response against tumors in other parts of the body. And some of the companies we've talked with have shown us data in mice where they've been able to do this. So they'll inject a virus into one tumor on a mouse, on the skin or somewhere close to the surface, and that will lead to the immune response kicking in and eliminating other tumors at other metastatic sites in the mouse.
And I believe that and please correct me, Emily, if I'm not right here, is that company that has shown really great, amazing results in the mice is going to be starting clinical trials very, very shortly.
I think that's correct.
Yeah, we actually have this trial open, I think, with another center. But there's, of course, with these type of trials, there's always a competition for slots. So.
Gonna see questions that we've got here. It's gonna be a little tricky to try to do this.
I would also put a plug in for the ALK vaccine approach, because the side effects with protein vaccines or dendritic cell vaccines tend to be, pretty minimal or mild. And while we are still finding out what that's going to do, for example, Dr. Lam has a vaccine study planned or in activation. Yes. And of course, the Mass General group has this vaccine in development, which is being activated as well.
I think one thing that's worth highlighting is that this is really an incredible time for cancer research. And with Dr. Camage here, Dr. Carbone, they really can speak to this even better. We're in truly the molecular age where we have the tools to gain insights into the sort of the underpinnings of ALK and why it's not working in terms of the TKI response, and to better characterize the immune cells that are in play. And then even more exciting is the fact that we have tools to actually then deliver therapeutics sort of as small sort of nanoparticles or as part of vaccines, like really sort of just biologic machinery within each of these sort of therapies that we've sort of touched upon. So I'm just really optimistic that we're going to be able to find a way with all of the different molecular technology advances that we've seen over the past 5-6 years.
Right. I think in this regard, COVID actually has helped us. So this idea of these messenger RNA vaccines, you've also heard about the idea of personalized cancer vaccine. So the question is, if we can find the peptides of the ALK protein that are stimulating the T cells in the immune system, you can actually code those on a messenger RNA and put that into an individual and then try to stimulate that response. That also means that if you can actually pull out a person's T cells, find out which pieces of ALK are being presented on those T cells or being sensed by those T cells. You can then make that personalized vaccine. And so there's that idea of the potential of a personalized vaccine approach. And I'd also mention, just in the general case, the idea of the T cell approach. When you take T cells out of a person, you actually then want to grow them up outside the person and put them back in. And there's a bit of art to that. It's not that immune cells are not like bacteria. They're not simple to grow. It's art as well as science. But it's still a great hope and a great possibility, this idea of taking out the T cells, your T cells that see the tumor, making millions of them and putting them back in. That's one of these approaches that is being arranged, worked through. And then the other is, of course, to put on T cell receptors on those cells that will recognize, again, the ALK protein or fragments of the ALK protein and target the immune system to the tumor. And so, and I also mention that because it's not going to be a one size fits all, I think, right? We're going to have a number of cards in our hand, and we're going to have to be able to pull out the right card to win the trick for each of us.
I think that's a great point. Truly personalized medicine, where hopefully one day we can do a series of tests on a particular patient, understand, okay, we need to do TKI plus TILs or TKI plus an additional protein degrader or some combination thereof. And I really look forward to that day.
So in case this conversation's getting too serious, I love this question. Dr. Erin Schenk talked earlier about the don't eat me pathway. So there was a question here. Wait, did she say the don't eat meat pathway?
As a very a proud meat eater, I did not.
So tell us a bit about the TIL research that you've been doing, again, in words that 9-year-olds are going to understand, or 10.
Yeah, so we grow up, billions of T cells from someone's tumor and then give them back to the patient, right? So this is like getting a big army put in, which will, in principle, kill off the residual tumor cells. But it does require some additional treatment with it, right? So you have to make space for this big army by giving 5 days of chemotherapy beforehand, which has its own set of toxicity to it, or side effects. And then, of course, some patients respond, some patients don't, and we're trying to understand why that is. We still have some patients that have had complete responses from being treated in 2018, but that's a handful of people, right? It's not nearly enough. Another interesting technology, like you mentioned, is the T cell receptor. And for those of us who have p53 mutations in our tumor, it's another mutation. There already are clinical trials which are targeting that mutation in trials. So you might want to know what your HLA type is, which is your white blood cell type, basically. And you may be eligible for a p53 T cell receptor trial if you have a specific type of mutation.
Does p53 actually cause cancer, or is it just something that makes the tumor more unstable so that it keeps getting new mutations more readily and it's harder to keep controlled?
Yes. Right. So TP53 is involved in part in DNA repair, but it's called a tumor suppressor. So the thing is, when it's working normally, it helps your cells behave well. When it's lost, your cells misbehave. And part of the misbehavior is in mistakes made in DNA. And every mistake in the DNA is another card that the cancer cell plays to try to evade your immune system, or to try to grow faster, or to try to go to a new place. Because we haven't talked about metastasis a lot, but obviously our tumors start in one place and then they go other places. And if we could block metastasis, at least we could deal with cancer at the primary site of its origin and not have to. That can still be lethal, but it's sometimes more manageable than when it goes to our liver or our bones or our brain. And so another area, but I think it's not as immediately being pursued by the community, is metastasis. I mean, there are groups that are trying to stop cancer cells from moving away from their site of origin. but it's a more complicated process, and it's even less well understood than the intrinsic behaviors of cancer cells themselves.
We have time for questions from the live audience, if there's anyone that wants to ask a question.
Here,
We can always count on Stephanie to get the ball rolling.
I'll roll. Sorry, joke. Yes, I'm gonna deal with the brain because that's something of big origin for me personally, which you all probably know. But my first question is, can T cells still be accessible from like older tissue? And maybe I missed that in the beginning. So like older tissue that was taken from your first biopsy that you might have gotten from your first TKI. And then I have two more real quick. Should I just say them all first?
I would say the answer is that in order to resurrect a cell from a tissue sample from long ago, you have to have treated that cell in a very special way, those cells in a very special way, and that's not generally done. So usually the cells are not preserved in a state where you can then put them in addition, cause them to divide. You'd have to intentionally take a core or take part of a block and store it in a special way, freeze it down in a special way, and then try, and you'd only succeed at some limited fraction of the cases to resurrect it. And it's got tens, if not hundreds of cell types in it. And so to find the T cells among those tens of cell types is another challenge. So that would be difficult or impossible.
But interesting, because my tumor cells that are in my brain are still ALK positive. So you're
So just to clarify, Stephanie, you're asking, can we find T cells for use as part of a TILS therapy, for example, as opposed to just profiling just to see if we can see T cells, right?
kind of, I guess, a mixture. Like if I were to be involved in the clinical trial, sorry, this is uncomfortable. Would it be something you would look in the past and start with those biopsies, or would it just be current only?
Yeah, we need a fresh, if it's for a therapeutic cell therapy trial, we always need to get a fresh either blood sample or a fresh tumor sample.
Okay, I'm sorry, I don't want to take up everyone's time. Brain T cells, can they be used and then injected with like your radiation? You were kind of talking on it, you almost touched on it.
Yeah, so brain tumors are tough because oftentimes patients are on steroids, right? Prednisone, when we take that tumor out, and those T cells probably aren't fighting fit to actually be able to be used for treatment. And there's not very many of them because they also are on steroids at the time the tumor comes out. So there hasn't been as much research in that area, but we have succeeded in culturing T cells from brain metastases, yes. But most of the trials out there will not consider that to be a usable source of tumor.
Note the STAT3 inhibitors that Mark was mentioning. It has been shown that that can potentially mediate a lot of the brain metastases that we see, not just in ALK. So, hopefully we'll be able to get one of these STAT3 inhibitors in trial and see if we can see that promise play out.
I have two questions for the panel. So the first is, Dr. Schenk, do you want to talk about your profiling in blood to give you a clue as to what cells might be involved? And if you do answer that question, you owe me $10 because you told me I had to ask that question. And then the other question for everybody is, we're in an ALK-positive session, okay? When we're talking about these clinical trials of immunotherapy, are they going to be done in conjunction with giving the TKI? And if the answer is yes, is that the way the trials are currently written? Or do you need to lobby for that? Dr. Schenk, you're up first.
I'll have to pay you later. So I love the excitement here about, the immune system and ALK-positive lung cancer, because I'm really excited and interested about about that interface too. It hasn't been well studied. We haven't really done significant amounts of work characterizing it. And I really feel like we're coming up to a point in time where we're going to try and better understand what is going on. How is the immune system seeing these tumors, and how can we capitalize on what's already the body is already seeing. And so in the study Dr. Kamich is referencing, we are taking blood samples at various times on patients who have started targeted therapy, TKIs, and looking at the immune cells within the peripheral blood and how they have changed, and associating it with how well patients do on that specific TKI. And some of the data we've been able to analyze so far shows that certain types of innate immune cells, and these are not the T cells, these are the burly bouncers in the tumor microenvironment, having lower amounts of those innate immune cells associates with certain patients doing better on TKI, having higher amounts of certain T cells, and these are really the effector cells, the cells that go in and kill the cancer cells, appears to associate with better outcomes as well. So some of these studies help to better understand, what does an immune system setup look like where a patient has a really good response to TKI? What is that substrate? What is that baseline already in a person's body? And once we have a better picture of that, can we help other people where their baseline maybe doesn't look like that? Is there a way to modulate those bad cells or modulate those good cells so that the patients who maybe are predicted to not do as well on targeted therapy can, by modulating the immune cells? So the TKI question, I think that's a really interesting question in terms of just mechanisms or how the immune therapies plus targeted therapies might work, as well as a question of safety in my mind. We know that some of the early phase 1 studies where immune therapy was really coming out, and we were excited to try and help all patients with lung cancer with immune therapy, where if we combine them with targeted therapies, and this was definitely in ALK-positive patients, as well as patients with other types of drivers who received TKIs, there appeared to be a higher incidence or more frequent side effects that happened that were quite severe. Several patients unfortunately died because of which died on these trials, probably due to some of the side effects immunotherapy and that specific ALK TKI caused. And so I think about it in two ways. One, in certain circumstances, it makes sense in my mind, background bias, tumor microenvironment, in immunology training, where I want to try to stimulate the immune system through two ways, meaning the effects that targeted therapy can have, as well as the immunotherapy, but also a concern about patient safety. So I think, all told, if these trials do move forward in a combination fashion, that we need to be really thoughtful about close monitoring, as well as taking various samples to understand what signals we might see if some of these immune side effects happen.
Yeah, and the other point there is just that we can use these TKIs, I think, in the current trials as a bridging therapy. So you get your tumor resected, and then you're in the kind of a holding pattern, waiting for that cells to be manufactured or grown. And that's a good time to restart, I think, your TKI pill. But then when you're in the post-treatment period, you're sick, you're wiped out, sometimes you're still in the hospital, and we're not going to be restarting the TKI in that setting. It's the biggest question, I think, to your point, is what do you do afterwards? The patient's responded pretty well, let's say, or let's say they've had a stable disease. Do you add back in the TKI? And that's an unanswered question. On the current trials, if someone were to do that, they would be considered non-evaluable once they start that second treatment.
Ben needs to catch a flight. He's already 3 minutes late. So can we give Ben a round of applause? Thank you so much for joining us. And if someone can help get him down to his vehicle and on his way, that'd be great. And we'll keep the panel going here for a few minutes. We're going to get Dr. Carbone started right on time at 10 o'clock. There's a question here. If I can turn it back on again. An oncologist hates the question. It's probably the question an oncologist hates the most. I'm going to look at Dr. Land here as well as Dr. Camage. And the question is, well, doc, how much time do I have left? They don't want to answer that question. And they shouldn't. And there's a question here, and it's probably the same question that researchers don't want to hear the question. What's the timeline for getting these available as normal treatments for patients? And researchers probably hate that question too, but obviously the answer is going to be different depending on whether you're talking about TIL therapy or TCR therapy or vaccines or oncolytic viruses or tumor treating fields or all the many different other technologies that are out there. But of the ones that you think are most promising and within reach of the clinic, what sort of time frame do you think that we're looking at for 2 or 3 of those different treatments?
Well, I think, in terms of the immune targeting therapies, there are several concepts that could be brought to the clinic rather quickly, and are already in development, like the vaccine effort that Dr. Awad and Dr. Chiarli are working on, as well as our group. And then the cellular therapies, those have already been in clinic for some time. Really, the question is part of the question is how effective are they for ALK specifically, such that we would want to prioritize those treatments for our patients at this time. So in that regard, I think, for instance, some of the cellular therapies, which I'm a huge fan of, I'm not personally sure that we're quite there yet in terms of having a cellular therapy that works especially well for ALK, such that we would prioritize that for our patient, for example, who's progressing on lorlatinib and that sort of thing. But they're already in clinic or should be in clinic, in the next year or so. In the meantime, we have wonderful trials by Dr. Dagogo Jack and colleagues who are combining ALK TKIs with drugs that are already approved for lung cancer, but not specifically for ALK, to address some of the bypass mechanisms. And then just briefly, the organoid effort from the Michigan team. We also have a small pilot of a similar effort where we can actually functionally try to identify why the TKI is not working as well and which pathway, maybe the bypass pathway like MET, that Dr. Camage and Dr. Doble have really sort of helped highlight. And so using functional tests like that may be able to help identify bypass pathways that we can address with off-the-shelf drugs, not just MET, but FLT3 and other drugs used for other cancers. So yeah, those I think are most readily accessible.
There's a few others too. In other disease types, there have been trials that are already open using inhibitors of myeloid-derived suppressor cells within the tumor microenvironment, or like IDO agents. So some of these things have already come into clinical trials. It's just a matter of thinking about, do we think putting patients with ALK-positive lung cancer would be helpful for them? Or is there a good rationale to try to explore that, for example? And the don't eat me signal, those antibodies are also in clinical trial right now for blood cancers. So, there's a possibility that we'll have some safety signals, and then we can just, consider the trials in the sense of efficacy. Could these potentially help patients with ALK-positive lung cancer? And I think a really interesting note in terms of the STAT3 inhibitor is some of the broader inhibition that STAT3 does also inhibits some of these suppressive immune cells. So while it might interfere with additional bypass pathways within the cancer cell, there might be an additional effect of remodeling the tumor microenvironment to make it more welcoming to T cells to come in and eliminate the cancer cells.
Terrific. We're going to have to wrap up pretty soon. One question again, probably going to try to need to make it understandable to people without a scientific background that this is someone that's pretty into the science. Is there any movement to employ an mTOR inhibitor alongside a TKI?
So mTOR is another signaling pathway. So I'd say this falls into the combination therapy bin of small molecules,
Yep.
right? So I think there are people who are interested in many different bypass pathways, and mTOR is one of them. I just don't know how active it is right now.
Terrific. Great. All right. I think at this stage we'll wrap it up, get Dr. Carbone up here on time. To me, I'm so grateful for every day. My signature, anyone that's gotten an email from me, they all know my email signature. It says treasure every day. And I really do. I'm very grateful for the TKIs. And I'm really excited for the future. Thank you.